adaptations that occur in response to exercise and that they are intrinsic to the working muscle. So this is the classic paper by John Halazzi, but I thought it was interesting when you were showing the Nobel Prize paper from 1953, Watson and Crick's paper. This paper was also being published in Nature, and it was essentially looking at myoglobin concentration in response to forced activity in rats and birds. So even going back then, we knew that the adaptations that occurred to exercise were essentially, many of them were mediated at the level of the mitochondria. So this slide has also been introduced earlier today, and essentially it's just to point out that it's a complicated process that leads to mitochondrial biogenesis. But mitochondrial biogenesis is one of the major molecular adaptations to endurance type activities. And there's been a sort of re-energization in terms of the interest in mitochondrial biology, both in terms of health and disease, and this has sort of been, I would say, stimulated by several papers that came out in the early, just around 2003, 2004, that essentially showed that that there was a downregulation of mitochondrial function associated with certain disease states. So my interest was in diabetes, but it's also been shown in aging, some forms of cancer, and also in obesity, which is basically what I've summarized really on this. Now, in terms of exercise and sports performance, we know that the mitochondria is the site of aerobic muscle metabolism. It's the major site of ATP resynthesis. And it's also, as I said, the major organelle that undergoes adaptation in response to exercise. In the case of disease, impaired function or reduced expression of OXPHOS genes, highlighted here is associated. I'm not going to say that it causes lifestyle-related diseases, but it certainly is associated with lifestyle-related diseases. Much of this, I think, though, is probably a consequence of physical inactivity. So there are sort of two seminal papers in terms of the description of mitochondrial biogenesis. I mean, there's been many reviews since, but this one appeared in 1996. This one appeared in 2001. And the reason I've highlighted them is because they describe the time course of adaptation to exercise in terms of mitochondrial content. And both of them really conclude that it takes about 6 weeks of endurance training to bring about a change in mitochondrial content in the muscle. But we know that the adaptation to exercise in terms of metabolic adaptation and also in terms of mitochondrial enzymes can actually occur with even about 3 bouts of exercise. So there were several short-term training studies that were done in the mid-1990s, and they pretty much showed that adaptation of the mitochondria occurs much, much faster than this 6-week period that's described to be required for a change in mitochondrial content. So by content we mean the actual number of mitochondria in the cell. What this review actually highlighted, though, was that within the first week of adaptation to training, there are changes that do occur in the mitochondria, and that's what we were interested in our proteomic study. So again, at the turn of the 21st century, there was a review paper from Ken Baldwin, which asked, researching the exercise sciences, where do we go from here? And there was a section in there that really got our attention, and it was really about the emerging fields of research, which were functional genomics and proteomics. So a recent review by Eric Hoffman, they looked at how proteomics has expanded recently in terms of the exercise sciences. And what they did was to go and look at a 10-year PubMed search, basically looking at for studies that dealt with proteomics in general. And so you can see that there was, pre the mid-90s, there was very little happening, and then there's been a sort of exponential increase. And we've seen that with several other disciplines within this conference. I went back then and looked at papers that specifically included were brought up by the search term skeletal muscle proteomics. And pre the year 2000, there was actually no paper that dealt with skeletal muscle proteomics. And then there's been a gradual increase in terms of articles per year over the next 10 years. However, if you look for studies that relate directly to exercise interventions and that look at the remodeling of skeletal muscle, there's less than 10 out there at the moment. So I sort of felt like the odd man out when I got asked to present on a proteomics paper at a genomics conference. So we're going to call this functional genomics from now on. And this phrase here that I've heard mentioned in our lab is that proteins rule the world, not DNA and not mRNA, but it's proteins that exert the physiological effects. So this is the study that we published last year, and I'll just take you through this for the majority of the rest of the talk. So what we had was a 14-day training program. So we were interested in short-term training, and the idea is that the most rapid phenotypic changes in terms of skeletal muscle actually occur within the first 14 days of the onset of an exercise training stimulus. So we had training sessions on each of the 14 days. These previously sedentary men trained for 1 hour per day at about 80% of their VO2 max. Pre-training VO2 max throughout. And we took biopsies at various time points, after the pre-training I should say, after the first exercise bout, after the 3rd, 7th, 10th and 14th exercise bout. And these biopsies were all taken the morning after the evening training session. So they were about 16 hours post-exercise. So in terms of the anthropometrics, the change that we saw in terms of percentage VO2 peak in the course of those 15 days was around about 14 days was in around about 15 to 20% increase in percentage VO2 peak. And just in terms of the training log itself, and I just want to make a small point. When they began the exercise training, it was at about 80% of their VO2 peak. But by the end of the training program, because we kept them at the same intensity throughout the training program, in actual effect, in terms of the overall stimulus relative to their post-training VO2 max, they actually were only training at around about 70%. So exercise, as you can imagine, became easier as the trial went on. And that may have implications for some of the responses that we saw in response to exercise. So again, just to show, this is just to graphically illustrated the difference in the change in VO2 peak. But I also just, based on the theme of some of the earlier talks yesterday, I thought I'd include just the relative increase in terms of aerobic capacity. And again, you see even in the small cohort of 8 people, the differences in terms of the gain in aerobic capacity in response to 14 days of fully supervised training. Everything was supervised and monitored by oxygen uptake during exercise as well. Very large differences in terms of the response. So the point I should have made was for the proteomic study, we only focused on 3 time points, which was day 0, day 7, and day 14. The data that I'll show where we have all 6 time points, this is unpublished at the moment, but it basically just corroborates a lot of the data the information that we saw in the proteomic study. So we saw no change in mitochondrial DNA to nuclear DNA ratio, so we suspect that there was no change in mitochondrial number in response to the exercise training bout. The classically observed change in citrate synthase activity was obvious after as little as 1 day, and then it plateaued after 3 days of exercise. And then we saw gradual stepwise accumulation in terms of the mRNA for COX-4, which is an electron transport chain component. And we also saw the same thing in response to protein, this increase in response to exercise training. So there are several recent reviews that discuss skeletal muscle proteomics, and I would encourage anyone with an interest in this area to read them. I don't have time to cover all the background now, but the model that we used for our proteomics analysis was what's known as two-dimensional fluorescence difference gel electrophoresis, so known as DIGE, 2D DIGE proteomics. Now, one of the advantages of DIGE proteomics is that it allows you to pick up very small changes in protein content. And the reason you're able to do this is because you use a pooled internal standard. So you pool basically all the samples that you have, you use them. And then you can compare a pre to a post in terms of the other 2 samples. So you essentially run 3 different samples on the same gel and using the same internal control across all gels, and it allows you to have this very sensitive measurement of changes in protein. So basically on the overlay then there's some software that allows us to identify differentially expressed protein spots, and from that then we get a peptide sequence and we can identify the protein that was changed in expression level. So this is essentially what a 2D gel map looks like. So it's separated in one dimension by molecular weight and separated in the other dimension by pH. And the eagle-eyed among you will notice that the pH range is finite. So there are some limitations in terms of 2D gels, that there are certain proteins that do not migrate into the gel. And just like the molecular weight is limited as well, we don't see all proteins of these molecular weights appear on the gel. So what we were actually able to identify was 800 protein spots that were that migrated into our gel. If any of you are familiar with mitochondrial biology, you'll know there's about 1,500 identified mitochondrial proteins. So we really were only looking at around half of the mitochondrial proteins that were able to be ran on a gel such as this. So this isn't exclusive, the results that we show. So again, this is just to illustrate that we basically have 3 different time points, and these are just 3 candidate mitochondrial proteins that we subsequently then validated by looking at their expression through Western blotting. So this is the readout that you get from the Decider software, and then we subsequently went and validated that both on mitochondrial extracts and also on whole muscle lysates. I'm going to show all of these proteins on a schematic on the next slide, so it's not important to read them all now, but the point I wanted to make was just to highlight these 2 columns here, which is essentially the fold change at day 7 and the fold change at day 14. And for most proteins there's an increase, for some there's a decrease, but generally speaking the range, the magnitude that we're looking at is somewhere between a 25% change as being statistically significant up to about 2.5-fold. So it's kind of a small range in terms of the amount of change of proteins in response to exercise training, but significant nonetheless. So how did this all look in terms of mitochondrial function? So I can start from clockwise, but essentially what we're seeing is major reorganization, or remodeling I should say, of the mitochondrial electron transport chain. So this seems to be happening at complex 1 and complex 5 in particular in terms of increases in protein expression, whereas we found this unusual finding, which was a decrease in COX5A. So if you remember on the previous slide, I showed an increase in COX4, which is kind of a classic marker of mitochondrial adaptation. One of the isoforms within the same complex was actually decreased in response to training. We saw increases in creatine kinase and adenylyl kinase proteins. This speaks to to alterations in phosphotransfer activity in the cell, basically providing more ATP at a faster rate. We also saw some changes in proteins that are associated with contractile properties, and some of you might be surprised to see that we saw contractile properties associated with mitochondria. But if you go into the mitochondrial literature, you'll find that many people who have looked at the proteome of the mitochondria and have used more sophisticated methods than us to isolate the mitochondria, generally find that because these mitochondria are tethered to various parts of the contractile apparatus, that you do tend to get contractile proteins in your analysis. So collectively, these changes indicated a shift towards a more more oxidative muscle fiber type. Then for the rest of the proteins, essentially what we're seeing are changes in the TCA cycle and the malate aspartate shuttle. And essentially, again, most of this seems to point to the classically observed observations of an increase in the ability to oxidize lipid, an increase in the ability to regenerate ATP, which are, as I said, classic adaptations to mitochondria or to exercise. One thing of interest here was a change, which was kind of why we were interested from the very start, was the idea of mitochondrial protein synthesis. And on the earlier slide, the one that looks at mitochondrial biogenesis, one thing that we know is that the protein synthesis machinery of the mitochondria is very complicated, but it's responsible for taking genes that are encoded in the nucleus and then somehow transcribing them and translating them into mitochondrial proteins and incorporating them into the mitochondria during biogenesis. So I'm going to come back to one of these just at the very end of the talk. So I think I've summarized most of this slide, and it's just to say that we see extensive remodeling of the mitochondrial protein, of the proteome, in response to just 2 weeks of exercise training. And again, just to point out that most proteins show increases after 7 days of training, with no increase after 14 days of training. But there are other proteins that show a time course dependency. And this just probably relates to the half-life of these proteins. So where could we go with information like this? So how can we use this hypothesis-neutral design to then be hypothesis-driven in terms of using this mitochondrial proteome information? So one of the proteins of the mitochondrial protein synthesis machinery is a small heat shock protein known as alpha crystalline beta. And what we found was this was downregulated by about 50% in response to training. So we were interested in what would happen if we actually silence this in muscle. So again, this is unpublished work and it's just very preliminary. But we were able to knock down this protein, this is at mRNA level. We were able to knock down this using siRNA in primary human skeletal muscle cells. And when we looked at markers of mitochondrial adaptation, not to our surprise, PGC1 was actually elevated in response to exercise, in response to this silencing, and there was also an increase in this component of the complex 1 of the electron transport chain as well. So again, the level of knockdown that we got was actually quite modest. So this is an experiment that we really should go back and do again. But this is just to illustrate that there are ways that you can use this, as I said, hypothesis-neutral data to come back and to look at with hypothesis-driven research. Another way that you can take this data is to look at something known as ingenuity pathway analysis. So this is a subscription-based computer program, and essentially what you do is you take your fold changes and you take your proteins that have changed in response to your intervention, and you use them as seeds for pathway generation. So again, this is hypothesis-free. It's done automatically by the program based on published literature and relationships between different proteins under different physiological conditions. So the ones that are appearing here in they're appearing here in yellow and grey in this case. These either were up or down regulated in response to training, and it then generates a network that you could potentially go back and look at. And of course, again, for those of you familiar with mitochondrial biology, one of the nodes that comes up as being potentially regulating the majority of these proteins is unsurprisingly PGC1. So just for the last, I mean, we would have measured this anyway, but it's just interesting that you can do this, and you could potentially go back and look at a variety of different proteins if that was your inclination to do that type of research. But just to point out what we actually went and did, I think, well, just very briefly, PGC1 is a transcriptional coactivator when it's overexpressed in muscle. Essentially, the muscle goes from being a mixed muscle to essentially a very oxidative muscle, shown here by a change in appearance in terms of being red. So PGC1, and this reflects the myoglobin content, so the increased mitochondrial content and oxidative capacity. So the question is really, what would happen if we went back and measured PGC1 in these samples? So we did that, and we measured at mRNA level, and we also measured at a protein level. And similar to the changes that we saw in terms of a time course response, we also saw a stepwise increase in the amount of PGC1 protein expressed in these muscles. The other major transcriptional partner that works with PGC1 in the control of mitochondrial genes tends to be ERR alpha. And so we saw, again, an increase in ERR alpha mRNA expression as training proceeded, and this was also reflected by an increase in ERR alpha protein after 3 days of exercise, which plateaued during the training program. And then one last final data slide is just to say that we also looked at the sirtuins. So this is a really interesting area of research at the moment, and the sirtuins being regulated by NAD/NADH ratios, and very popular because of the new research that's come out on resveratrol. So we looked at SIRT1 mRNA expression and SIRT1 protein expression, and as you can see, the increase in SIRT1 occurred in a time course dependent manner as well. We also measured SIRT3 protein and NAMPT, which is a regulator of NAD levels in the cell. The point I just want to make on this, I don't really want to get into the discussion of sirtuins, but just to point out that if we look at something like mRNA level and its relationship to protein level, I think SIRT1 is a good example of a note of caution that I want to make. If you were to have measured SIRT1 mRNA only at one time point in response to an exercise training regimen, and it happened to be day 14, and you saw no change in mRNA levels, then you might conclude that SIRT1 is really doing nothing in response to exercise. And this happens quite a lot in the literature, where people will publish only on mRNA expression rather than looking at the downstream protein. However, if you look at SIRT1 protein expression, and again, if we taking the same example, if we looked at just day 14, there's about a 2-fold increase in SIRT1 expression. And as I said, we haven't done the mechanistic studies to look at what is happening, or what is this a cause or a consequence of exercise training. But the point being here that only looking at one component of the flow of information in the cell, such as mRNA, doesn't really give you a full picture of what's going on at a molecular level in response to exercise. So the majority of what I've said there is summarized there. There's some information that I didn't show. One thing that we're seeing coming up is we think there are early fission events that are occurring. So some of you may have noticed a decrease in mitochondrial DNA at the start. after one bout of exercise. This was an unusual response. We think it's a stress response to the first bout of exercise, and it seems to be related to an increase in expression of DRP1, so a fission event occurring in the mitochondria. Some information I didn't get to show as well was a decreased expression of something known as CD38, and this is a key enzyme in the consumption of NAD levels within the cell. So one thing that we're seeing is an increased expression of NAMPT and a decreased expression of CD38, and we think that that's regulating NAD levels at rest in muscle, and that's somehow altering the sirtuin response and consequently the adaptation to exercise. So just in conclusion, I'll just thank most of the collaborators that were responsible for a lot of this work and the funding agencies that helped out along the way. So that's it. I'll take any questions.
Okay, so the floor is open. If we have the mic somewhere. We have one just down at the front here, please, Kathy.
Thanks for a beautiful talk. You looked at a number of different pathways, and I guess I think of the AMPK pathway, the PGC1 alpha pathway, and the calcineurin pathway in terms of switching towards the slow myogenic program. And I'd really like your insights as to whether you think all of these pathways act in concert, how they, or in concert, is that however you pronounce it, or whether they act independently, how they interact with each other, because you are certainly showing, you know, for example, upregulation of phosphorylated AMPK, as well as your effects on PGC1 alpha.
Well, it's a complicated answer. There are a lot of actual crosstalk between the pathways. So, for example, AMPK is one of the upstream AMP kinases is CAMKK, which is a calcium kinase. So CAMKK is activating both AMPK and calcium CAMK2. So there's some crosstalk there. We also know that the effects of AMPK seem to be related to sirtuin metabolism as well. So they were some papers that came out from Johan Owerks's group. So there's some crosstalk there as well. I think in relation to whether they're all acting in concert, it's hard to answer. But in studies that have knocked out either CAMKs or if they've knocked out AMPK, you still, by and large, get the same exercise response in terms of gene expression. Some of the metabolic things like lipid oxidation and glucose transport, they seem to be some things are happening there, but in terms of the exercise response at mRNA level, most of those tend to be conserved in those knockout animals. So I think there's a lot of redundancy within the pathways.
We have another one here, Cloud.
Very nice talk, thank you. In your model, I think you have shown a few times that there was no difference between day 7 and day 14. So that puzzles me because it suggests that we may have reached maximal adaptive response in a week. How do the figures that we know from the muscle of athletes or people who have been doing this for years, how do the value differ from those that you observe after 14 days? Have we reached maximum responsive after 14 days?
So I don't think we have, and I think it's an artifact of the experimental design, because if the point I was making on, if you look at the training log, for example, we train them at the same intensity throughout the 14 days, and I think people adapt very, very quickly to a stimulus of exercise training. So what should have really been done is that they should have had their VO2 max assessed after 7 days and then reassigned a new higher level of intensity throughout for the next 14 days, and I would like to have seen that done in a future study. I think what's happened there is that the training stimulus was insufficient as training progressed. One thing that hasn't been done, and I would again like to see this done, is that there have been studies that have looked at the transcriptional response pre and post training. And what they always will find is that the post-training transcriptional response is less than pre-training. But it's always done at the same absolute work rate as absolute power output as the pre-training. So I don't think that's actually measuring the same thing. You're essentially asking someone to do a less intense bout compared to a more intense bout, just because they've adapted to the training protocol. So I think what would be nice to see is if you could reproduce or repeat the post-training exercise bout at the same relative intensity rather than the same absolute intensity. I think that might answer the question a bit better.
Thank you, Dr. Egan, for this nice presentation. It's been quite a long time that we know that not always RNA mirrors protein, and yet you have shown here clearly for a certain one. So, but we are still using RNA. How do you foresee the future in this field? What do you see if you had to choose what to do?
I think in the case of acute responses, I think it's transcriptomics is still very useful. I think in terms of the utility in training studies, I would have my doubts. But you have to remember that transcriptomics is very, very sensitive. And proteomics, one of the major drawbacks of proteomics is that the technology isn't near as advanced as it is in terms of being able to examine the whole proteome, as opposed to the way transcriptomics can examine the whole transcriptome. So I think in the case, my interest is in proteomics, but I think you have to acknowledge the limitations. And again, the point I made was that in the case of mitochondrial proteomics, we were only looking at 800 out of the 1,500 mitochondrial proteins. So there could be so many other things going on in terms of training adaptations that we don't know about. So I think the thing is to combine both. I mean, maybe it's sitting on the fence a bit to answer like that, but I think the thing is to combine both.
Okay, thank you.